I did invite a couple of my colleagues who worked on this with me. One or
great.
two. One might
Excellent.
turn up. Yeah.
Excellent. That's okay. I'll keep admitting people in as we go, but it's 6:30 South Australian time, so we'll get started. And I'd just like to acknowledge that this Butterfly Conservation South Australia National Science Week event is being held on Kaurna land, where I am, and we respect our First Nations elders, past, present and emerging, and respect that their knowledge of the land and the care they've taken over many millennia. And we also acknowledge that they never ceded this land. So thank you very much, everyone, for coming on to the Butterfly Conservation National Science Week event. I don't know whether you're aware, but this National Science Week goes from the 14th to the 22nd of this year, and there are something like 397 local events and 236 online events and 14 competitions that you could join in. So we're one of many, and it's our great pleasure to have Professor Mike Kearney from University of Melbourne here to talk about his research, and I will just introduce him in a moment. Those of you who don't know about the Butterfly Conservation Association of South Australia, we've been in an incorporated organisation of amateur lepidopterists and people who really appreciate butterflies and moths since 1998. And it was first formed because people were asking the question, where have all the butterflies gone? And so we have developed quite a lot of resources. We have two wonderful websites, one a more general public website called, the URL is butterflyconservationsa.net.au, and the other one is a more research focused, great deal of detail by one of our Life members, Roger Grund, called sabutterflies.org.au. Our two main resources are two books called Attracting Butterflies to Your Garden. We're in the second edition, 2016, that was published, and Caterpillars, Moths and Their Plants of Southern Australia, which was published in 2019. So we have an online shop, and if you're interested in joining the association or getting any of our resources, you're most welcome to go onto that shop. We, as a committee, we chose the family of Nymphalidae because it is the largest, and then we went around looking for a speaker or speakers to actually tell us a little bit about the family. And in my brief understanding of the brushfoots or the Nymphalidae is that there are over 6,000 species across the world, and you know, we have quite a few of those in Australia. But I'd just like to introduce Mike, and Mike can tell us a little bit more about his research and then his presentation. So Michael is an ecologist and evolutionary biologist. His research focuses on limits to where species can occur, especially in relation to climate. He uses a combination of field and laboratory research and theoretical modelling to study this problem. His hands-on work is mostly on insects at present, particularly grasshoppers, and hopefully we'll get Mike to talk about grasshoppers to us at some future time. And he's worked extensively on reptiles in the past. He also works collaboratively on birds, mammals, including humans, and frogs. His group spent a number of years working on the common brown butterfly project. He hasn't quite finished yet, but we're going to hear some of that work. So over to you, Mike. Thank you very much for joining us.
Pleasure. I'll just make sure I share the right screen. You see a title slide?
Yep.
Yeah, thanks, Jerry. So I must admit, to start off with, I'm no expert on butterflies in general. So I have an appreciation and love of butterflies, and have some very fond memories of seeing my very first blue Ulysses as a I don't know, 9-year-old or something up on Dunk Island on a family holiday. But my real expertise in terms of natural history knowledge is with reptiles, and then more recently with grasshoppers. But I guess I started off as a natural historian as a child, and sort of hunter-gatherer sort of instincts. I wanted to catch lizards. I wanted to find more, and I was interested in where to find them and why I was finding them in some places and not others. And now basically I just keep doing the same thing and get paid for it, trying to understand what limits where species can and can't occur. And I've been trying to develop some very general ways of asking this question: what limits where species can and can't occur? And I did some work on a nymphallid, on the common brown, in this context, and I'm going to share some of that research with you. And I've called this talk "How do species with complex life cycles respond to climate?" because butterflies have very complex life cycles, and it makes for a more challenging problem, but also a very much more interesting problem than some organisms with much simpler life cycles. So from a broad scientific perspective, they're an interesting challenge, and it's interesting to think of the consequences of making your life cycle complex for how you respond to environmental change. But also I'll hope to give you a little bit of more natural history knowledge about this very common and important butterfly as we go along. So the whole project started out because of Paul Sunnock's, a colleague. We've been talking about these sorts of issues, and Paul thought, why don't we try and do a project on the common brown? Paul's a geneticist, and he thought the common brown would be an excellent species to look at because he's also close colleagues with Neil Murray from La Trobe University. And Neil had worked in the past on this species with his PhD student Kay Pearce, and they did a big study on genetic variation using sort of the old school genetic approach of allozymes. And they had all these specimens that had been kept from back then, apparently, or they were all in the museum somewhere. And so the idea was to use some of those old specimens and revisit some of these questions that they got a little bit of an insight into back then in the early 80s. So we applied for some funding to do this and got the funding, and that was back in 2007. That's when the project started. And so I'm talking about things that happened quite a while ago. It was actually quite fun revising all of this stuff. And as Jerry mentioned, I am still working on this, trying to get sort of the final answer to this this problem of what limits this species. And you'll see a tentative answer at the end. Yeah, it turned out those old specimens had been thrown away, so that was a shame. But nonetheless, we decided let's tackle this problem and try and understand what limits this species. The Heteronympha genus has a number of different species within it. So what you can see in this figure here, this is all taken from the Atlas of Living Australia. So you'll see a few wayward points that may not be correct, but that's what's currently in the Atlas for all the Heteronympha specimens. Some of these have come from iNaturalist, some of these are based on specimens in the museums. And then, so within that range, we have all these different species. We have the wonder brown, Myrifica, which hugs the east coast quite tightly. And then we've got a number of other species that are sort of just generally southeastern in distribution. So the shouldered brown, Heteronympha penelope, the spotted brown, Paradelpha, Banks's brown, Heteronympha solandri, which has a sort of more narrow range and moves up into the high country a bit more. Heteronympha cordace, the bright-eyed brown. And then the common brown, which is what we're focused on here, which occurs throughout southeastern Australia, obviously comes into South Australia, also occurs in Tasmania and in southwest of Western Australia. And there's various little dots out here, some of which are real. This is real, so this is up in the Flinders Ranges, and in fact there used to be a population out in Mudawinji Gorge apparently, and this is real too. This is Carnarvon Gorge. So why does this species distribution look the way it does? What's stopping it from going any further than where it is now? And if we can answer that question, we can also answer how will it change as the climate changes, which is a very important problem for us all. Now we're going to go way back to a couple of South Australian scientists who really shook up the world a bit in ecology in terms of understanding the problem of what limits the distribution and abundance of species, particularly animals. So this was Herbert Andrew Arthur, also known as Andy, and Charles Birch. They were working at the Waite Institute in South Australia and basically tackling problems to do with pest insects. And Andrew Arthur was an ecologist, generally speaking, and Birch was a geneticist, but an ecologist as well. And they wrote this book, The Distribution and Abundance of Animals, and it was an interesting book because at the time there were all these fancy population models and models about how you get something called density dependence, where a species starts to increase in numbers and then the predators or the parasites start knocking it down again, and you end up with these stable populations. And they took they had a whole lot of data rather than just models, and they said, look, if you look at some of the species we're studying, like the small plague grasshopper, Ostracis cruciata, actually these things, they disappear, they go extinct locally, and they live in a metapopulation, and they were the first to really introduce that idea of metapopulations, where it's not like the species abundance goes down, and then the pressure's taken off and they come back up again before they go extinct. Sometimes they just go extinct, but because they're scattered across the landscape and there's connectivity, then you get the species persisting through the landscape. They talked about distribution and abundance being different aspects of the same problem, and they used this little grasshopper, which was a pest that I was studying, the small plague grasshopper, as a way of illustrating this point. So you can see the range of the species in blue there. And they said, if you take a transect through a species range, like this grasshopper, this is a gradient that we've got here from wet and cool through to dry and hot. And you might find in a certain part of the range in the middle, numbers are quite high. So if you had a plot of, this is that transect in kilometers through the transect, and this is time, and this is abundance, then you'll see the fluctuations are happening, but they're not really getting that low. If you take another point in the sort of maybe the cooler part of the range, where it's getting a bit cooler and wetter, the numbers will be fluctuating and the overall abundance will be lower, but they're still there. Whereas if you go somewhere quite marginal near the edge, you'll see every now and then they're there and then they're not. So this is the local sort of meta-population dynamics. And really the distribution limit is where the abundance tends towards zero. And so in that sense, distribution and abundance are related. So they said, look, if we're analysing this problem, what limits the distribution and abundance of species, there are 4 things we need to think about. One is the weather, and they were really emphasising how important the weather can be for controlling the distributions of many species. And they saw massive fluctuations of the insect populations with the weather. The second thing is food, availability of food. And this relates to the climate as well, the weather conditions in terms of how that drives the available plant material for animals to eat, and then also for that may be a direct constraint if they're eating plants, or an indirect constraint if it's insects or something else that's eating the plants that that species of interest is eating. So food is second major important thing. And then 3, other kinds of animals or animals of the same kind. So interactions within the species, with each other, competing for food perhaps within the species, and also those interactions that the theoreticians of the time were fascinated with, predation and parasites and that sort of thing. And 4, a place in which to live, which is shelter, appropriate shelter sites. So they just boiled down to these very simple kind of natural history perspective things that we need to understand. And so this is the way I do like to. I like the way these 2 think or thought, and I've been trying to basically apply this in a way, or build on this in the work that I'm doing, centered around the concept of the ecological niche, which Andrew Arthur and Birch didn't really use, but then the niche concept goes back way back to 1917 with a fellow called Joseph Grinnell, who described the niche of the California thrasher, a bird that he just described all the different conditions that this thing requires to be able to survive, and called that the niche. Then an ecologist who was an aquatic ecologist called George Evelyn Hutchinson came up with a more sophisticated idea of this, where you actually quantify it as a volume in environmental space. He called it the n-dimensional hypervolume, which is a very fancy sounding name, but basically said, well, we can characterize the range of rainfall conditions a species can occur under. We could characterize the temperature conditions, and then we have the combination of those that are suitable. And we could perhaps take that to a third dimension with, say, soil pH, and we've got a box. And then we could add a fourth dimension, which I'm not going to draw because it's physically impossible, fifth dimension, and so on. And then we can characterize where in environmental space is it suitable for this species, and then predict where else we might find it. So, and he had this distinction between the realized niche, which is where the environmental conditions where the species actually occurs, and this abstract concept of the fundamental niche, which is if you took away all the predators and the parasites, how much broader would that be? And so you're only considering the real physical constraints. So what I've been trying to do in my research is find ways in particular to characterize this fundamental niche idea. The very bare basics of what an organism needs for survival, what it needs in terms of the right temperature, the right level of moisture, humidity, enough time and food to get through its life cycle, avoid the bad, stressful times, and make sure we reproduce at good times, and get through the sensitive periods of the life cycle when maybe you're little, just hatching or an egg or whatever ends up being the most important part. So trying to characterize that the real fundamentals of temperature, food, nutrition, water, I should say, on where species can occur. Now, since all of these past scientists I've mentioned were working, we've ended up with some really powerful environmental data sets that we can use to answer this question about particularly how climate limits where species can occur. And that's through what are called climate services. And Australia really led the way in the development of climate services. What they do, so you can see these two maps show the temperature and rainfall stations that we have in Australia. And you can see we don't have them everywhere, but we might want to make a prediction in places where we don't have a weather station. So how can we predict what the temperature would be in those places? So that was, that problem was solved by Mike Hutchinson from the Australian National University. He came up with some statistical methods to actually smooth across the landscape what the rainfall and temperature and other variables would be. And I actually asked Mike, why was Australia the first to get these climate services? And in fact, because the technique he used, he got from someone from the US, but they weren't using it to interpolate their weather stations and make these climate services. And he said, well, in Australia, we've got these big gaps. You can see these big gaps in rainfall, where rainfall weather stations are, and obviously the temperature stations are also even more sparse. Whereas in places like the UK and the US, they had stations everywhere, so they didn't feel the need to make these surfaces. So it turned out that we ended up with in Australia developing these techniques and having the first climate surfaces. And so from then on, all of the sort of modelling approaches that use them have kind of started off in Australia just because we started off with these layers. And there are two ways that people use these layers to predict where species can and can't occur. And the first way is a correlative way. And it's the way that most people use these layers. And so what these models do is they start with the distribution points that you can get on the Atlas of Living Australia or somewhere like that. You say, all right, here are all the places where my species occurs. And you get the temperature and rainfall and so on in all those places. And you use a descriptive statistical model to characterize that space. And so, the simplest possible one is you say, oh, what's the range of temperatures and what's the range of rainfall? And then you say, where else do those combinations occur on the landscape? And you can make a map and see where else the climate's similar. And that's a super powerful approach. It's been used extensively. It's one of the biggest areas of research in ecology at the moment, and it's really powerful, but it's also limited, because when you make this kind of a model, it's correlative, it's not causal, it's correlation rather than causation. And so there are so many possible ways that temperature and rainfall could be causing a limit on a species. And the model somehow captures those processes through the statistical relationship, but it doesn't let you unpack that model and find out what those processes were. It's a black box. You can't see into it. You don't know what processes he captured. And also, so that's not satisfying in that, you know, for me, I want to understand what's limiting rather than just be able to predict. But the other real problem is that it's a statistical model and it only works within the combinations of the conditions you use to make the model. As the climate's changing, we're getting new combinations of environmental conditions. And so these sorts of statistical models are no good for making these predictions because they don't know, they haven't got any information about what that is like in the future, what would happen to that species, because they're not causal models, they're correlative models. So the alternative approach is a mechanistic approach or a causal modeling approach, where you take some kind of a model and you try and use that to, which is basically being explicit about processes. You say, here are the things that I know limit this species, and you explicitly build those in, and then you try and make a prediction to the landscape and see whether or not you've found something limiting. And so what I'm going to show you is how I've tried to do this using the common brown butterfly as an example. And if we can do it, we get that pleasure of actual scientific understanding of what's actually limiting a species, but we also have a much more powerful way to predict what will happen to them in the future. So what is a mechanistic model? What do you have to do? So in the previous, with the correlative model, what you start with are the distribution points, like the atlas records. With a mechanistic model, you start with the things you measure about the organism. So that's an important distinction. So, but also you need to know what environment the organism actually experiences on top of that. So one of the first things you really need if you're going to make a mechanistic model of a species and how it's constrained by environment is not the climate, which is what the climate services are giving us. You want the microclimate. So climate is measured, and weather is measured by weather stations, and say the air temperature is measured in a weather station by a box like this called the Stevenson screen. And it's white to shield it from sunlight. It's ventilated so the air can move through. But most importantly, it's about 1.5 meters above the ground. And the point of that is that you want to get the air temperature of the region, not the air temperature of that particular place. And the closer you go down to the ground, the more the temperature gets influenced by the things around the box. The further you go up, the more it's representing the general atmosphere in the area. So that's why weather stations are put up high, but organisms live down near the ground. So a little caterpillar on some grass, this is a Spinifex Grassland, similar to what's behind me in my screen, my Zoom background. And this is a visual image, and this is below is an infrared image showing the temperature an infrared camera would see. And you can see the thermal variation is going from about 32 degrees to 56 in this one snapshot of time. Under this bush, it's about, under this Triodia tussock, it's about 32, whereas out in the open on the surface, it's about 56 at the hottest. So that's one challenge. We need to be able to predict microclimates. And I won't go any further into this, but that's something we do. We have a way of turning this information we have on the climate into the conditions actually experienced on the surface or underground. And it's not just the air temperature, it's how much sunlight's hitting the animal, how fast the wind's blowing, what the humidity is, all these sorts of things. And then we need the model of the organism. And this is what I was saying before. This stuff, it's all quite heavy. It took me a long time to understand, especially as an ecologist who didn't really study a lot of maths or physics or chemistry, thinking that I didn't need that to be an ecologist. But what I've ended up having to learn is a fair bit of chemistry and physics and coding to put it all together. And I'm basically taking this from what other people have done and sticking it together to be able to make these calculations for an organism. And it's the physics of heat exchange. So you have a heat budget. It's the physics of exchange of water. You have a water budget. And it's the physics of metabolism, which is basically thinking of the organism as a chemical process, taking up things and building its body and making babies. So the mechanistic model that I've been working with does all of these calculations and basically allows you to work out how hot the thing will get, how much water will it lose, can it forage, is it warm enough to forage, all those sorts of things. And we put this into the context of a microclimate and we incorporate how the thing behaves Does it seek shade? Can it go underground? So it's involved, but it's possible to do, and it's possible to make good predictions that match what organisms actually are seen to do really well. And once you've got that, then you can say, well, what's the life history of this organism, and how does that change through time? So you can start the animal off as an embryo, as an egg, or as a, in the case of this sleepy lizard here, it's inside the mother. And so the embryo develops, and then it starts growing, and it matures, and it starts reproducing. And that gives us the trajectory of that life cycle, and we can see how long will it live, how quickly will it mature, how many babies would it have. And from that information, we can then work out, all right, well, what's the population growth rate, and go from the individual and how it's interacting with its environment to its trajectory of its life to the population dynamics, and ultimately make some prediction about the distribution. So that's what this whole mechanistic niche modeling approach is attempting to do. And it's hard to do, but even if you fail, you learn a lot along the way, because it's forcing you to look at it in a very complete way, and it makes you look at things you may not otherwise have considered, that things that may end up being really important that you haven't really, that you may, through your own biases, have not looked at. And so, yeah, it's a way of doing what Andrew, Arthur and Birch were talking about, incorporating all of these things. Really, I mean, you can bring in the other animals bit with this, but mostly I'm not bringing that in. It's basically weather, food, and a place in which to live. And, if we're doing this with some simple animals, like the sleepy lizard has possibly the simplest life cycle. The mother has a baby who grows into something similar. But the common brown, we have a complex life cycle. So now we get to a butterfly. Sorry, it took a while to get to a butterfly. This is the life cycle of the common brown butterfly, and it's an interesting one for a butterfly, where it's an annual life cycle. So in April, this butterfly is laying its eggs, and those eggs, they're developing, they hatch and they develop through 5 different larval stages through the winter, and they're feeding on grasses like native grasses like kangaroo grass and poas, but they'll also eat panic felts and other introduced species. So they go through their life cycle, and then they pupate. So that so they're starting off as an egg, and then they've got this mobile stage, of course, that's feeding, and they're nocturnally feeding. And then they go into the pupal stage, which is another completely different, environmental situation, and it doesn't have any behaviour. It just sits underground. They go underground a little bit and pupate. And then we, of course, have this incredible thing that emerges that is able to go into very different environments, has a much different, much greater dispersal capacity. And that happens in November and December, with the males coming out first, and then the females. And they mate. The males are chasing the females around in the early summer, and then you stop seeing the males so much, and the females sort of disappear as well in most parts of their range, until around about March, April, when it starts getting green again, and they lay their eggs, and the life cycle starts again. So that's the life cycle of the common brown. And you can see, trying to do what I just said with building a model of this whole thing, it's much more complicated with a species like a butterfly. So you've got all these different issues to think about when you look at that whole life cycle. There's when do you lay your eggs? That's really important in terms of what environment the little caterpillars end up in. You lay your eggs early versus late in that season, they may encounter different temperatures. Well, they might encounter warm temperatures at the wrong stage, or cold temperatures at the wrong stage. They may be experiencing a situation where there's no food if you don't get the timing right. So you've got the development and the growth rate and so on, and the possible stresses that they may face depending on when those eggs are laid. And then you've got when the pupa emerges, or the butterfly emerges from the pupal stage. And there's, what are the conditions that those butterflies emerge into? Are they able to survive all the way through to when they need to lay their eggs again? So that's what we're going to try. That's what we tried to do, characterize all of this for the common brown and see if we could predict where it can go, where it can live. And so the first part of this project was done by an honours student, Nat Prisco. And so, when I was saying before, we might be a bit biased in what we decide to study in a particular project. Well, we were originally thinking, let's look at the butterfly, the adult stage, but we ended up deciding actually probably a lot of the action is going to be with the less interesting eggs and larvae. So we started there and started measuring what temperatures can these eggs and larvae tolerate and how fast do they develop at different temperatures. So that's what Nat did. So this is an incubator. Nat reared hundreds and hundreds and hundreds of caterpillars from eggs and looked at different temperatures to see how development rate changes with temperature. And so that's what this first plot shows for an egg. It shows the development rate and the temperature. And she did 8 degrees up to 25 degrees. And you can see what you expect is the rate should go up and then start to come down again as things get a bit nasty and hot. And so you can see these eggs, they develop, increasingly fast as the temperature goes up. And there's a little bit of evidence that it's going to start to come down again as it gets to starts approaching 30. This is the first instar caterpillar and a very similar looking pattern. But what we found with the later stages, the later larval stages, is that as they get to the later and later stages, they become less and less tolerant of warm conditions and sort of less sensitive to temperature. And in fact, we didn't get any caterpillars surviving all the way through at 20 degrees, which sounds like a pretty nice temperature, but that's too hot for them to go all the way through. So remember with the life cycle I showed you, as these caterpillars are going through later and later instars or larval stages, they're going deeper and deeper into the winter. And what it seems is that this species is adapted to cooler and cooler conditions as it goes through to later and later stages. And it really can't handle being warm when it's a late stage larva. So that was very interesting. Often we just assume that we measure the thermal response at one stage and assume that all the other stages are the same. That's not the case with common brown. It varies depending on what stage you're at. Nat also fed these grasshoppers leaves and measured the area of leaf that they consume in a certain amount of time at different temperatures, and was able to get the way that feeding is constrained by temperature. And you can see this graph here shows a third instar caterpillar, and you can see that it's feeding faster and faster as you get warmer, and at 25 it's sort of optimal, but 35 it's actually feeding pretty well as well. And this is the stage that's still in the relatively warm time of the year. But when you get to the final instar, the fifth instar caterpillar or larval stage, they're really having trouble at 25 degrees, and 30 degrees is terrible for them. So they're very sensitive at that late larval stage to high temperature. So that was very interesting to discover. Looking at the development overall and comparing the males and the females, remember I said the males come out a bit earlier, and when you measure the development rates in the lab, the males go faster. So this is the duration of the larval period, and you can see at 8 degrees, the females take 225 days to complete their larval stage, whereas the males are taking less or fewer than 200 days. And that's consistent across all the 3 temperatures she looked at. 15 degrees, you can see that the effective temperature here on development time as well. If you take caterpillars down up to 15 degrees from 8, you're going from 225 days for a female to develop to about 160 days. So massive effective temperature on how fast they complete their life cycle. Interestingly, the pupal stages, they didn't really differ between males and females at how fast they went. But again, a very, very strong effective temperature. And the body size, this is sort of interesting that the mass of the females changing through time. The females actually emerge and are much larger. They emerge a bit later and are much larger than the males. So, on top of the complexity of the life cycle, we've also got males and females doing something slightly different. And with that information, Nat was able to say, well, what would we predict the length of the life cycle to be if we theoretically lay the eggs at different dates? That's what she's done here. She's laid the eggs in February, March, April, or May. And so this is showing the day of the year. And these bars, so this is laying the eggs in February, and this is the first, second, third, fourth. No, that's the egg, first, second, third, fourth, and fifth larval stage, and then the pupal stage in black. And so you can see as you shift the date at which they are laid a little bit later and later and later, you can see the corresponding length of time it takes them to emerge. But you can also see how much time they spend in each of those different stages. And the point is that it's quite variable. If they lay the eggs quite early, then they spend very little time in the first larval stage, and the second and the third and the fourth, and they spend most of their time as a fifth, and also quite a while as a pupa. Whereas if they lay their eggs in May, they sort of spend about equal time in all the different stages. And you also see that they don't, if you lay eggs a month later, it doesn't translate to a month later that you emerge, because there's a bit of a catch up going on by the pupal stage, for instance, because it's more sensitive to temperature than the larval stages. So it's really complex and interesting. And she was able to then say, well, what if we make it warmer, like the 2070 conditions, and was able to show the whole thing was shifted earlier to by about 30 or 40 days or so. And we went a bit further with this. I've put this life cycle image back just so that, just to emphasize that when you start your eggs really determines how these other different stages experience their environment, and that could have positive or negative effects. And what we had a little bit of information on from museum specimens of when emergence was occurring over the years around the Melbourne area. And there's a tendency for emergence to be about 1.5 days earlier per decade. So there's a decline in, or an earlier and earlier emergence as time has gone on in the common brown around Melbourne. And the air temperatures around Melbourne in the time window of April to October, when these things are developing, has been going up and up and up. So putting all of, these are all the curves that Nat got from doing the incubation experiments, putting all that together like I just showed you, we got the dashed line here and the white open squares, which was similar to what we saw in nature about the butterflies coming out earlier and earlier. So it seems to roughly work that we can predict that order of magnitude change in how early they're emerging from this physiological information. And in this particular paper, we teamed up with a climate scientist, David Caroli, and he did some analyses which are represented by this particular plot here, showing how much warming is happening per decade. In reality, it's 0.15 degrees per decade. He showed what is the chances of that happening if there wasn't human-caused climate change occurring. And so these bars are basically showing what would happen in the extreme of a whole lot of simulations of the climate without greenhouse gases. And it shows that you can totally attribute the climate change that's happened in Melbourne to humans, and then you can attribute therefore the earlier emergence of the butterfly around Melbourne to climate warming by humans. So that was kind of interesting. Then I had a PhD student, Maddie Barton, also work on this butterfly. She did a whole range of things, but including looking at geographic variation in this butterfly and seeing whether what we found around Melbourne actually holds in other places. And just briefly, one really interesting thing she found was when she measured development at all these different places, you can see represented by the different coloured dots. This is the larval development time at 15 degrees and 20. And in both temperatures, you can see this purple one is going faster than all the others. And the purple one is Carnarvon Gorge. So right up in the north, the caterpillars develop faster at the same temperature than in the south. So not only do they vary in how they respond through the different stages of their life, but different populations are also different in how they respond. One interesting thing that she did was to look at how body size changes across this whole range of temperatures that occur. So these are the winter temperatures that occur throughout that whole range from up in Carnarvon Gorge down to Hobart. And the thorax length for males in the open circles and females in the black, no response. They're just the same body size if you measure them in Tasmania as if you measure them in Carnarvon Gorge. But if you incubate them at different temperatures, at 15 degrees compared to 20 degrees, the 15 degrees are much bigger than the ones at 20. So we don't really know why that's the case. Why in the lab, they're very sensitive in terms of size to temperature, whereas in the wild, we don't see a signal of it. So that was a lot of work on the eggs and larvae, but also we did some work on the adults, and Maddie did a whole lot of work on what temperature do butterflies get and what's the role of the wings, which has been a question people have been interested for a long time with butterflies. You know, there's this idea that they open their wings and use it as a solar panel and they focus the light on the thorax and warm up. So you can see some images here of the sorts of experiments that Maddie did to try and get further with this problem. There's a butterfly in a wind tunnel with a light shining on it. And you're blowing wind at a known speed and you're warming the butterfly up and letting it cool down, and you can measure all sorts of physical things about that. And you can pin the butterflies with different angles of wing and look at what is the effect of the wing in the end. And you can see we measured the temperature with a camera that is infrared, and you can see the different, this is a butterfly with its wings closed and open and partially open. And the short story is that actually the mostly open but not fully open is the best. But it seems like it's very complicated how butterflies' wings affect the way that they exchange heat. And the main thing is that when they've got their wings open, they're allowing the sunlight to hit the thorax. But if they position themselves so the wings are blocking the wind, they can get a bit warmer. And so the wings actually blocking the wind flow, direction of the wind flow, is probably one of the main reasons that the wings are helping them get warm, rather than acting as little solar panels, magnifying heat onto the body. So that was also very interesting. And she showed that we could then predict the temperatures of the butterflies in nature pretty well. That's pretty much what this graph shows using this model, which I won't go in much more detail into. She also did some funny experiments trying to work out the sensitivity of these butterflies and their ability to fly at different temperatures. I say funny because we were trying to work out how do we set up a way to change the temperature quickly and see what the endurance is of the butterflies. And we came up with the idea after experience sitting in a tent when camping, how hot the tent gets. We thought, well, let's set up a tent and let's fly butterflies around in a big tent and let the tent warm up with the weather. And that's what Maddie did. And you can see these two graphs on the left show the flight duration in minutes of males and females as a function of the ambient temperature in the tent, where it was getting really hot. It was 50 degrees at the hottest. And you can see these butterflies are flying around at 50 degrees. So it's not like they conk out at 40 or something. And I do remember on Black Saturday, I was hanging out at my parents' place. It was 46.7 degrees in Melbourne that day. And in the hottest part of the day, I went for a walk around their house. So they live in a nice, cool area. But it was still, it was 46 or something in ambient air temperature. And there was a common brown flying around their carport in that weather. So this is the range of temperatures, though. You can see it is dropping off in endurance as the temperatures get too hot. But they're also flying as low as about 15 degrees. The females even around just above 10. And then this is what you see, the proportion of individuals or number of individuals flying at their field site, given the predicted body temperature. And as it gets up to about 25 degrees, that's where it seems to be optimal for them to be flying. So we characterized the heat side of the adult part of the butterfly's life cycle. But Maddie also characterized a whole lot of things about the metabolism and that side of things with the butterflies. So she put them in the lab and she fed them different amounts of food, and she looked at how many babies they have. One experiment she did was a little bit cruel, but it was looking at survival of these butterflies with access to food and water, water but no food, or no water or food, no food, simulating what sorts of environments they may actually encounter depending on when they emerge. And this was done at 20 degrees, 24 degrees, and 28 degrees. And you can see they can live as long as 80 days at a relatively low temperature with food and water, but the survivorship drops as the temperature goes up, and that's simply because they're living at a faster rate. It's not like because it was stressful, but you warm up a butterfly, it will live for a shorter amount of time because it's running faster. Its metabolism is running faster. But you can see that without water, and certainly without food and water, you see the life cycle, but the lifespan is very much reduced. A lot of butterflies, of course, just They don't feed a lot of moths. They don't feed at all, so they're just relying on their stored reserves from the pupal stage. And so the common brown can rely on those reserves for about a week or so, but not much longer. So they really need to find food and water. So we could put all that stuff Maddie did into a model of the whole energetics of the life cycle and metabolism. And I'm not going to go at all further into this, but know that there is a way to do it. It's really complex and hard, but we worked out how to do it. And it's called dynamic energy budget theory. But what it allows you to do is start as an egg and run that model through, going through all the different stages, all the way through to how long can they live, given the temperature as a butterfly, what is the lifespan of the butterfly as an adult, given how warm it is. So you can go from egg all the way through to the end of the life cycle. And so, getting to the culmination of this, we can put it all together and get a trajectory of the butterfly's life cycle in the form of a graph like this. This is a particular example for Melbourne. And what you're seeing on this graph is a year, calendar year, and it's the mass of the butterfly being predicted. And we're starting as an egg, and in fact, the egg's really starting in April here. So the egg develops, and then you've got when it hatches here. This is the duration of the first larval stage, second, third, fourth, and fifth. So you can see the body mass is growing, especially large amount of growth in the final instar, the final stage. And then this is pupation. So what happens during pupation is the animal's losing body mass and converting itself into a new form, and then it hatches and is laying eggs. So that's the trajectory in Melbourne under a certain environmental condition of living out on some grass that has full sun on it. Okay. And what you can see, this black line below is how moist is the grass. And so where it drops lower than this sort of horizontal line here, it's actually a bit too dry for. So that's a bit too dry in late January, early January, sorry. That looks like February actually in this particular year, and like around the beginning of January, late December in this particular year. And the model's accounting for that. If they are a larva at the stage where there's no food, then the larvae are going to start starving, and the model's predicting whether they'll die or not, whether it's too long between food to survive. And this even more complicated looking graph below is showing all the sorts of complexities we're accounting for in the model when we're making these predictions. So the yellow is the body temperature we predicted the animal to have all the way through these different times. And that's using the microclimate and doing a bit of heat transfer physics. The dashed blue is what we would have predicted if we just assumed that their body temperature equaled the weather station air temperature. And so you can see that the eggs are getting much hotter in the summer. The butterflies are actually cooler than air temperature in the summer, as a weather station would have measured because they're thermoregulating. The amount of shade that they need is this green area. So you can see part of the reason why these butterflies in the summer aren't getting too hot is because they're using shade in the model. And you can also see the depth of the animal in black. And so they're only allowed to just go a little bit into the bushes, into the ground as a larva. But as a pupa, they go right down to about 15 centimeters and they develop down there. And then as an adult, we allow them to go down holes and things. And that's what common browns do. They'll go to shady places, so they can even go into caves and things during the summer when they're waiting for the rain to come for the grass to lay their eggs on. So you can see there's a lot of complexity we're building in here, but we're able to put all that together and say, what does the life cycle look like and do they succeed in getting all the way through it? And we can do that for multiple years. So this is a plot for 1990 through to the end of 1995, showing in all these different years, when we run the simulation in Melbourne with full sun on the grass, no problem, they survive. What if we go to somewhere in inland New South Wales, go to Dubbo? Not so good. You see what happens is, in this first year, the larvae hatch and there's grass. You see these lines sort of horizontal here with maximum moisture in the grass. And they pupate, but then they don't quite make it through pupation because actually they did have a bit of trouble for whatever reason in the model, and they weren't able to finish pupation. there's one particular year they were able to get all the way through the life cycle, and that was obviously a relatively wet year, where the timing was just right in terms of the moist grass. But if we make it so they lay their eggs in the shade and the larvae are growing up on shady grass, oops, there's no problem, except for a couple of years. So this is telling us that we would expect in a place like Dubbo, and I would also expect in a place like Wilpena Pound, where the common brown occurs in South Australia, that they're going to have to be in really shady places. They're not going to be able to survive out in the open. Here's Carnarvon Gorge in the shade, and a similar situation to Dubbo. So as long as they're in the shade, they're doing fine and they're able to survive. And that's what we see in Carnarvon Gorge. They do survive there. But say we go to Alice Springs, computer says no, can't live there. So we can do this across all of Australia and make a map and say, where would we expect the distribution limit to be? And this is what I'm sort of building up to here. So what this map shows you, we're incorporating all these things I was talking about before about the timing of events, and can they run the gauntlet of the environment and get all the way through to their life cycle? And in 2005, with this particular calculation, it's showing where this failed, and all the coloured areas are where this failed. All the white areas are where it succeeded. And the colour coding shows why it failed. In these areas, the high alpine areas in southern Tassie, they failed because they got too cold. In the red areas, it failed because they got too hot above the lethal limit. In the brown or orangey areas, they ran out of food. And really interestingly, in the grey areas, the females didn't survive long enough across the summer to bridge the summer and get to the green time to lay their eggs in the autumn. The next graph shows the stage at which they died. So I just said that the old age situation, that's for the adults in purple. So you can see that the grey matches all the purpley sort of colour. But you can see also like the egg stage is where you're getting a lot of heat stress in some of these inland areas. So we can, with this model, be quite specific and develop quite specific hypotheses about what is the problem in these different places causing the butterfly not to be there. And it's not just one reason, of course. It's a whole lot of possible reasons that we're seeing is limiting the butterfly from being in different parts of Australia in that year, 2005. We can do that for lots and lots of years. So here's a series of maps, 2005 through to 2008. And if we put them all together and say, where are the places where they could always survive in all these simulations, you get all the grey dots, all the grey squares. And matches, well, pleasingly well. We were amazed how well it matched actually with the actual distribution, which are the brown dots. So this is preliminary though, like there's some issues with this, the way this was done that I'm fixing up, but you can see that it is possible to put in all the complexities of the life cycle of the common brown and actually come up with a prediction that actually matches somewhat with its actual distribution. And allows us to understand what sort of mechanisms are causing problems for this butterfly in different parts of its range. And then we can say, all right, well, what if we just added 3 degrees to the climate? And you can see in red is where they would disappear, in green, where they would expand. And that, apologies if you read green colorblind, that's not the greatest choice. And grey, where they would just persist. And so knowing these mechanisms, we can better understand how to conserve them. For instance, we might realize that it's actually the need for shade, and we need to create shady environments for our butterflies in certain parts of their range. Whereas in other places, it maybe there's not much we can do because it's just getting so warm that the adults can't live long enough to survive through the summer to lay their eggs in the autumn. So I hope from this talk, you've got some idea about how we can make these kind of calculations for any kind of organism really, but in particular, how we can do it for a butterfly. And I hope along the way, you learn a little bit more about butterflies. So thanks for that, and very happy to answer any questions you might have.
Thanks very much, Mike. We've got 3 questions on the chat that came up during your talk. So Bruce, would you like to unmute yourself and ask your couple of questions?
And Jerry, shall I turn off the presentation or leave it up there?
I'll leave it up
Yeah,
there.
okay.
Hi, Mike. Thanks for that. That was a great presentation. Very interesting. I was just wondering if you could shed any light on what might happen to the females during the February to March period. You said that they sort of disappeared from the scene.
Yeah,
Any thoughts on that?
Yeah, and it varies, depending on where you are in Australia. So I think if you're in Tasmania, you'll probably see them flying around because their problem in Tasmania is it's cold and they're having trouble getting through their life cycle in time. But in Sydney, for instance, I think people see them in the sandstone cave areas. They congregate in shady, possibly caves. I've seen common browns around holes in shady places around Melbourne. So I think what they're doing is they're surviving off their stores and just staying somewhere where it's cool and humid, so they're not losing a lot of water and food. They might be lucky enough to be somewhere where there is nectar in it, but in other places I've seen them hanging around some tree that's flowering in the summer, and so they're in the shade of the tree and they've got food and water. But it's possible that they can actually handle a certain amount of time without food and water as long as they find somewhere cool and moist to hide.
Okay, great. Thank you. Just wondering too, in relation to the observations on the size of the adults, would that be you mentioned, I think, that at 20 degrees, the body size was somewhat larger than at 15 degrees. Do you think that would be like a surface area to volume issue where the larger adults are more able to withstand temperature changes better than smaller ones?
It could well be. It actually goes the other way. So the warmer they are, the smaller they are. And that's a general thing that people see in a lot of organisms. It's called the temperature size rule. No one really understands why it happens. So yeah, it would have all sorts of consequences because body size affects so many different things. It would definitely affect how warm they get, but it might also affect how long they can survive. Like the bigger you are, the longer you can survive without food. So yeah, and it's very, very interesting that despite that really strong effect we saw in the lab, we don't see it in the field. So it suggests that they've got ways of, maybe, there's obviously there's a lot of variation in microclimates, like I showed you on that slide of the desert scene. And if the females are choosing where they lay their eggs and so they're making sure that the babies are somewhere where they're going to be able to be warm enough, or the pupae, I mean, it's probably mostly the pupal stage where that's happening. And so the pupae might be able to control where they go. And so they're actually, going somewhere cool enough that they're coming out at sort of optimal size, even though there's a huge variation in temperature across that distribution. Their behavior sorts it out and they're able to, because I agree, like, it would also affect flight, of course, the dynamics of flight and their ability to fly. So there's probably a lot of selection on them to be the right size and to avoid that effect of development temperature. The other complication, of course, is that they were constant temperatures that we incubated them at. And in nature, it could be quite variable depending on how deep the pupa is.
Thanks for those great questions, Bruce. Claudia's got a question about the sexing the caterpillars, Mike.
As in, how did we do it?
Is Claudia still online? Do you want to ask your question, Claudia? If she can't, I'll ask it. Just got another couple of messages. Okay, Claudia's question, Mike, is how were the caterpillars in that study sexed? Can you determine sex in the larval stage, or do you have to wait for eclosion?
Yeah, and I'm pretty sure we had to wait for eclosion. This was done in 2008, and I'm stretching my memory back. I remember, it being a problem, and I'm pretty sure that's what you had to do. There probably is a way to tell, like there's probably a genetic way. You could possibly, use the malts, but we didn't have the money to do anything like that then. Yeah, I'm pretty sure we had to wait and see what we got.
Yeah, certainly there's quite a lot of detail in Roger Grunn's research on our sabutterflies.org.au about the adult sexing is really easy, but he doesn't make much comment about the caterpillars or the eggs as to whether you can actually define the sex.
Yep.
So sorry you couldn't unmute Claudia, but yeah, look, are there any other questions from the viewers? It's been a fantastic presentation, Mike, and really appreciate the time you've given to us tonight.
No problem.
Okay, if there are no other questions, I'd like to thank you on behalf of Butterfly Conservation for giving us a very enlightening talk, particularly when we've got another question just coming in, particularly when we've just had this IPCC report saying that we're in diabolical trouble. We really need to do something about it. So hopefully we can respond appropriately. There's another question from Julie. Does fecundity change with ambient temperature?
The overall speed with which they lay their eggs, the frequency does, but not the total amount. So as they get warmer, they'll lay the same number of eggs in a shorter period of time if you hold everything else the same. But that's if they've, if the temperature change doesn't affect their ability to find food and so on. So it could be that as it gets warmer or colder, they might have trouble getting the resources to produce their eggs. So that's where you could have an effect of temperature on fecundity, but it's not directly through temperature itself. All the temperature is doing is making them run faster, which is weird to think about because we're always at the same temperature. It's funny to imagine what our lives would be like and our culture would be like if our body temperature could fluctuate and we could shut down to 15 degrees and go to sleep over the winter or something and avoid the Melbourne winter. But yeah, for these ectothermic animals, temperature is just the speed of their bodies is just going up and down all the time with their body temperature.
Yeah, we, as I mentioned earlier, we've got a citizen science project with the Iridomyrmex ruffiniger and the Lucia limbaria, particularly the caterpillars, but we obviously monitor the butterflies as well. And it's really amazing to be able to see the temperature, particularly during summer. We will have to go out there at 8 o'clock to be able to observe the caterpillars because of the ambient temperature. The ants just bring the caterpillars up straight away so they can come and feed. So
oh, really?
It's really a temperature response. It's quite amazing.
Yeah.
Anyway, look, thank you very much, Mike. It's been great meeting you online, and we do hope you enjoy reading our 2 books. And certainly that invitation, it will certainly stand for giving us a talk on grasshoppers or anything else that you might like to talk to us about, especially if you can come over to SA face-to-face next year. That'd be really great.
Yeah, I'd love to come back to SA, and yeah, thanks for the books. They're great. I'm going to enjoy looking through them. Okay, bye.
Thanks everyone for joining in, and the event has been recorded, so we'll do a little bit of editing and then we'll put it up on our YouTube channel so that other people can enjoy this great science. So thanks again, Mike, and thanks everyone. Stay safe.
Bye.